All right, then. Today we're going on a bit of a journey. Inside your own body, actually. We're on the hunt for some tiny, tiny powerhouses that, well, make everything possible.
Have you ever really thought about it? How on earth does your body break down a big meal in just a few hours? If you just left that same food on a plate, it would take ages, literally years, to break down on its own. So, what's the body's secret? Well, the answer is that you have this incredible fleet of microscopic machines whirring away inside you all the time. They're called enzymes, and honestly, they're the unsung heroes of, well, you. They're the ones running the whole show, making the chemistry of life happen at a speed that's just mind-boggling. Okay, so to really get our heads around what these enzymes are, we need to start with a slightly bigger idea. We need to talk about catalysts. So, what on earth is a catalyst? Imagine you're playing matchmaker for two friends. You introduce them, get the conversation started, and then you just step away. You made it happen much faster, but you're not actually part of their new friendship. You're completely unchanged, ready to go again. That's a catalyst. It's something that dramatically speeds up a chemical reaction without getting used up itself.
And enzymes? Well, they are the body's very own super specialized biological catalysts. They're made of protein, and they are absolutely essential for pretty much every single reaction that keeps you going. We're talking everything from digesting your lunch to building new bits of DNA. They can build things up, a process called synthesis, or they can tear things down, like in digestion. They're your body's personal construction crew and demolition team.
All right, so how do they actually do it? How do these tiny little workers pull all this off? Well, the secret, it's all in their unique shape. It's all explained by something called the lock and key model, which is a brilliant analogy. You see, every enzyme has this very specifically shaped area called the active site. Think of that as the lock, and the molecule it's designed to work on, which we call the substrate, is the key. And just like a real lock and key, only the one specific key can fit into that lock. This incredible specificity is absolutely crucial. So let's walk through it. First, the substrate, our key, slots right into the enzyme's active site, the lock. For a moment, they're joined together in what's called an enzyme-substrate complex. Then, the enzyme works its magic. It might break the substrate into smaller pieces, or maybe help it join up with another molecule. And once the job is done, it releases the new products. And here's the brilliant part. The enzyme itself is completely unchanged, totally ready to do the whole thing over again. It's incredibly efficient. But, just like any finely tuned machine, these enzymes are a bit fussy. They need the conditions to be just right to work at their best. If things aren't perfect, the whole operation can slow down or even grind to a halt. And when we talk about conditions, we're mainly looking at two things, temperature and pH. For both, you see this classic pattern. Every single enzyme has its sweet spot, an optimum point where it works fastest. As you can see from these graphs, the activity climbs up to a sharp peak, but if you go too far in either direction, it just plummets. And guess what? That peak temperature for most of the enzymes in your body is right around 37 degrees Celsius. Sound familiar? It should. It's your body temperature. And that's no coincidence at all. Your body puts a huge amount of effort into keeping you at that exact temperature precisely because it's the perfect working environment for all its tiny engines. So, what actually happens when things go wrong? What if the conditions stray too far from that perfect sweet spot? Well, that's when you get system failure. And we're not just talking about a slowdown here. We're talking about the machines actually breaking for good. This breakdown has a name. It's called denaturation. What happens is that if it gets too hot, or if the pH is way off, the enzyme's active site, the lock, literally changes shape. It gets warped, and that change is permanent. And if the lock changes shape, well, the key doesn't fit anymore. The enzyme is rendered useless.
Now, this is a really important distinction to get right. High temperatures cause permanent damage. They denature the enzyme and it's game over. But low temperatures, that's a different story. Getting cold just slows everything down. The molecules get a bit sluggish, so the reaction rate drops. But the enzyme itself isn't damaged. Its shape is still perfectly fine.
Here's a great way to think about it. Putting an enzyme in the cold is like putting a factory on standby. All the machinery is still there, ready to go when you warm it back up. But exposing it to too much heat, that's like setting fire to the factory. The machinery is melted and twisted out of shape. The damage is permanent. Okay, so we've talked about the theory. We know that heat breaks these enzymes for good. But how can we actually see this for ourselves? Well, luckily, there's a pretty straightforward lab experiment that shows all this in action.
Right, here's what we need. Our substrate is going to be starch. Our special tool is an enzyme called amylase. Its one and only job is to break down starch. And to see what's going on, we'll use an indicator called iodine. Now, iodine has a neat trick. It turns a dark blue-black color when it's mixed with starch. If there's no starch around, it just stays its normal brownish-orange color. So, step 2 is simple. We add our enzyme, amylase, to the starch solution, and we hit the stopwatch. The race is on. The amylase starts munching away on that starch immediately. Then, at regular intervals, say every 30 seconds, we take a tiny drop of our mixture and test it with iodine. At the start, of course, it'll go blue-black because there's loads of starch. But as the amylase gets to work, you'll see that color getting weaker. The big moment we're waiting for is when we add the iodine and nothing happens. It just stays that orangey-brown color. That's our finish line. It means all the starch is gone. And what does this all tell us? It's beautifully simple. The faster that blue-black color disappears, the faster the enzyme is working. So by doing the same experiment at a bunch of different temperatures, we can actually plot out one of those graphs we saw earlier. We can find the enzyme's perfect sweet spot and prove the exact point where it gets too hot and just stops working entirely. So there you have it. From digesting your dinner to building your very being, these incredible little engines are working away nonstop. They are, quite literally, the difference between life and just being a bag of chemicals. It really makes you stop and think, doesn't it? What other amazing, invisible forces are out there, quietly running the world all around us?